Again, I'm going to talk about, I added a Q just to clarify things. So the talk is supposed to be about next-gen sequencing as a key step towards personalized medicine in this disease. And I'm going to talk a little bit about the data, but the data hasn't changed in like 5 years. So I'm going to get a little bit metaphysical also, because I think we're at that point where we got to get a little metaphysical with this disease. These are my disclosures. Um, so those of us who might know us, and Jamie's not here. So Jamie Chaffin and I are colleagues at Memorial, and she and I have a number of commonalities. We did residency together, and we have young families. And so when we found out we were coming here together, we naturally talked a little bit. And so on Monday of this past week, she and I were talking about cell phone reception. It's a little snippet of our email conversation. I had asked her at 5:13 p.m. on Monday, do our phones work in PR? To which she answered less than a minute later, it's the US, so I think so. Now, those of you who know Jamie, there's like a 50% chance that this is her way of saying, Paul, you're an idiot. And in fact, I do think that's what she meant. But in my defense, I had made an assumption in the question that I asked. There's always assumptions in questions that we ask. And the assumption on my part was that Puerto Rico's pretty far away, even if it is in the United States. It's about far away as Cancun, Mexico, and you don't get coverage over there unless you're roaming and get charged extra. So I thought it was a reasonable question to ask. We'd also talked earlier that day about the weather in Puerto Rico, and the question I asked was, how cold is it in PR? She said, it's in the 80s, and I said I better take the sweaters out of my suitcase then. And of course, the assumption at this point was that Puerto Rico is not that far away. It was in and is in the 40s and 50s. We are wearing sweaters in New York City, but it turns out it's actually pretty far away. And in my defense, again, the linear distance sort of belies the actual distance if you imagine it in the globe and all that other kind of stuff. And so there's an assumption that is present when we talk about next-gen sequencing as a key step in this disease. And that assumption is that single-agent oncogene-directed strategies that have worked in lung adenocarcinoma are also going to work in squamous cell lung cancer. In other words, squamous cell is the pork to lung adenocarcinoma's chicken. Now, we can get at this by taking a look at some of the higher level genomic comparisons between the two diseases. And so in lung adenocarcinoma, we're all pretty used to the fact that mutual exclusivity in our driver oncogenes is common. We knew this during the time of more limited genotyping efforts. We know this in the landscape of more complicated genotyping efforts, as with TCGA. And so I've listed in OncoPrint format the most common actionable driver oncogenes in lung adenocarcinoma. And you can already tell looking at the OncoPrint, if you're used to looking at OncoPrints, that things are fairly straightforward. There's not a lot of complexity that's there. And so if you were to draw boxes around the cases that were mutually exclusive of one another, it's pretty common. So the majority of cases are mutually exclusive, even for KRAS mutant lung cancer, certainly for EGFR mutant lung cancer, and ALK rearranged lung cancer, et cetera. And of course, the idea is that mutual exclusivity is the key thing that allows us to visualize lung adenocarcinoma as a pie. If it were not mutually exclusive, then we could not construct a neat pie. And of course, this pie works. Pie-directed therapy means that we have lots and lots of drugs, and apparently another drug, for this disease subset, either FDA approved for lung cancer or for other malignancies that we can provide. If we take a look at the TCGA squamous data, and again, these are the top, in this case now, putative driver oncogene events in the disease that are very different from lung adenocarcinoma. You can already tell just from a bird's eye view that things are more complicated. And if you were to draw boxes around the mutually exclusive cases, you can see that it's the minority of cases are mutually exclusive. And so you can ask, well, isn't this just the fact that squamous lung cancer is more complicated than lung adenocarcinoma? We always say that this is the case. And again, we turn to the TCGA data for mutations per megabase for squamous cell lung cancer, which is pretty high up there at about 8.1. But the idea is that the mean number of mutations per megabase in lung adenocarcinoma from the TCGA data is also pretty high up there at 8.9. So on average, this actually does not hold. Now, the sentiment is not wrong. It's just that the idea is that there is a subpopulation that is considerably less complicated in lung adenocarcinomas by and large than in squamous cell lung cancers. These are never smokers, and I think the best source of evidence from a rigorous high genome level standpoint is that Cell publication that Govindan had from a few years ago, where the never smokers that he had sequenced had about 10 to 20-fold fewer SNVs than the smokers in his series. And of course, never smokers are also more common in lung adenocarcinomas, anywhere between 15 to 20%. Again, these are TCGA data for the pie charts. The 4% background never smoker squamous cases tend to be a little bit more complicated from a pathologic perspective. Are these really squamous lung cancer cases? Or are they misdiagnoses? There's a little bit of uncertainty that's there. Nevertheless, even if they were from never smokers or pure squamous from never smokers, the proportion is much higher in lung adenocarcinomas. And the other part of this, of course, is that the driver oncogenes that we're used to targeting lung adenocarcinomas are concentrated in the never smokers also. EGFR, ALK, HER2, RET, ROS1, and now MET tend to be more common in these patients. And so if mutual exclusivity is uncommon in squamous lung cancer, does it make sense to make a pie? And this is one version of the pie for squamous lung cancer that you can construct. And so this takes the sort of standpoint that Puerto Rico is not far away, assuming that New York is lung adenocarcinoma. And so you create pie-directed therapies. This is circa 2015. This is the update to LungMap. We'll have another update later this afternoon or tomorrow afternoon or evening from David Gandera's presentation at World Lung, where everyone gets foundation testing, and then there's rational selection for targets based on what your genotyping ends up showing. One concern is that some of the early phase trial data that we've presented and published show that there's some modest benefit to single agent inhibition for the FGFR1 amplified subgroup, which has been sort of the leading target. Two drugs have had modest responses at 8 to 15% confirmed partial responses. And then for the upstream PI3 kinase aberrant patients, it's at unconfirmed 3% for the overall response rate and a PFS that's no better than docetaxel as a second line therapy. And so this takes the tack that Puerto Rico is actually pretty far away from here. But the idea is that Puerto Rico is exactly 1,606 miles from New York City, right? So there is a truth that's there that tends to get obscured based on assumptions and on relativism, and this is the metaphysical part. So the notion that next-gen sequencing is a key step toward individualized medicine in this disease, again, the assumption is that somatic alterations reflect the state of oncogene dependence in the tumor. Now, that's either wrong or it's right? It's one or the other. And so somewhere in there is that 1,606 miles that we've got to figure out, we've got to find. And so what are we missing? Well, so there are some probable things that we might be missing. The complexity of the disease we already went through. On top of that, there's some concern that adaptive signaling mechanisms might be in play. And I think BRAF mutant lung cancer as a paradigm for this, which I'll show in a little bit, is one way to take a look at this and one thing to be concerned about in the backdrop of complexity. And then there's some uncontrollable things that we have to deal with. Tumor heterogeneity for us is very difficult to control for in targeting oncogenes. And flat out being wrong, of course, is uncontrollable, because if you're wrong, you're wrong. And so in terms of adaptive signaling and complex signaling, Pirolito and Neil Rosen's lab published a pretty elegant paper in Cancer Cell a few years ago, taking a look at V600E dependent cancers. And the idea is that there's a steady state of high MEK and high ERK output that through a number of mediators leads to suppression of upstream RTK signaling. And so this engenders this RAS independent state that we're used to for BRAF mutant lung cancer and other cancers. Upon inhibition of BRAF V600E, though, with vemurafenib, for instance, or dabrafenib, you get shutdown of ERK signaling and relief of this feedback inhibition for RTK signaling. And so the receptor tyrosine kinase can signal, and they signal through RAS and through alternative RAF isoforms. You get heterodimers with CRAF, for instance, which then leads to a new steady state RAS dependent signaling. And when this steady state ends up developing, either early on after V600E inhibition or later on, will give the semblance clinically of primary resistance or acquired resistance. And we know that this is important because the strategies that have targeted this have worked. We know this from a phase 2 study of dabrafenib and trametinib. In this disease, the single agent response rate to dabrafenib, similar to vemurafenib, is about 32%. But when you add MEK inhibition, you get up to 63%. This is now an FDA-approved regimen for melanoma, of course, breakthrough designation for BRAF mutant lung cancer as well. And so can next-gen sequencing direct therapy? For those of us who say, yes, it can, there's a lot of additional work we need to do. We need to do better jobs at phenotype to genotype analyses, these N of 1 experiences, these outlier responders that we see in trials. We've got to pursue combination therapies, not blindly, though we need better models. And at this point, given the complexity of things and the failure of our existing preclinical models to translate into something that's worked in patients, PDXs, I think, are going to be the largest source of something that's going to be tractable. And this does not mean that large single-agent trials like LungMap are obsolete. These, I think, are going to be very important as discovery platforms that are not treating just a handful of patients, but many, many patients to find out exactly what's going on with these responders as well. And for those who say, no, it can't, then there are other tools that we have to try to begin to use to determine pathway dependence, things like proteomics, expression profiles, and again, a reliance on PDXs to probe both genetic and epigenetic phenomenon in this disease. Thank you. That's it.
So we have time for a couple of questions for Paul.
I guess I'd say two things about the gene list that you put up there. And I'm one of the authors of the 2CGA squamous paper. I really don't think there's a lot of evidence that PTEN is really statistically significantly mutated in lung squamous cell carcinoma or any squamous carcinoma. I think we continue to want it to be, but it really isn't. It doesn't come up on the significantly mutated gene list. Chromosome 10 is almost never has homozygous deletions. And then for the FGFR amplifications, if you actually look at the FGFR amplicon, it's actually barely expressed at all. So that's the other, I think there are some. And then the third candidate to mention is that the PIK3CA mutations, I think we really have to respect the heterogeneity of these mutations. The head and neck paper is showing us that there's a lot of mutations in the helicase domain. I mean, those are probably different than the mutations in the kinase domain. So I think we really need to think about these mutations as separate events.
Yeah, these are all important points and things that we've addressed before in terms of that fall into that bucket of whether or not these things are actually, oncogenic events. For the FGFR1 story, you're absolutely right. So there are a number of concerns in terms of picking these up, not through sequencing efforts, but through FISH, for instance, in terms of whether or not R1 itself is amplified. Overexpression of protein happens in a subset of these cases, and RNA expression as well, which is why the Colorado group is taking a look at these alternative biomarkers as response platforms to ponatinib in squamous lung cancer. I think the PI3 kinase story, I think you're exactly right. There's the semblance of events that happen potentially with greater frequency in squamous cell lung cancer, but whether or not these things, again, reflect for instance, absolute PTEN loss or loss of function in the setting of PTEN or hyperactivation of PI3 kinase signaling, for instance, PI3 kinase or PIK3CA amplification. These are things that probably in many cases don't end up happening, but have been sort of the low-hanging fruit, right? These are things that we can probe for that we've tried to build in as biomarkers, which have clearly failed, at least in terms of the BASIL-1 trial, which raises the concern about exactly whether or not these two major apparent high frequency aberrations are actually fueling oncogene addiction in these cancers.
Thank you, Paul.